It's beyond a thrill to be speaking here at the Broad. It's certainly been this mythical place on the other side of the world I've certainly heard plenty about. Yes, I create animations, and it's actually quite appropriate. You're probably all feeling very tired, wanting to have a little rest, and you're probably a little bit hungry too. That's actually one of my key audiences that I try and reach. It's my job to grab people like that. They've come home from work, and the kids are yelling, and dinner's trying to be prepared. The news comes on, and it's my job to explain something like breast cancer in 15 seconds or 10 seconds on a national news bulletin. Was initially the target audience of my kind of work and my kind of approach. There turns out to be, and I think I really want to say this to all of the community, there is a vastly bigger market out there for this kind of material. There's a huge demand for it in so many different types of audiences. And don't underestimate the demand and the need for this kind of work to explain all of science to the general public and to school kids and to university students and so on. A few of you, I think more than a few, have seen my shtick before, so I'm sorry you'll be seeing it again, but hopefully I've mixed up a little bit. And also I will be including some new works that are currently in progress that hopefully you'll find interesting, and we'll see how we go through that stuff. So the four areas I'm going to go through: 2 minutes on the tradition of scientific visualization, because in 2 minutes you can cover it. I'll be doing malaria, the malaria life cycle, which is one of my own personal projects that I spent about 12 months doing, and it is complicated biology, but yeah, actually I should say up front, the idea behind my work and what I've discovered, I'm adamant this is the way to go, is that when you create a visualization, particularly for the public, you never ever dumb it down. You don't simplify it. You show them exactly what the science is about. But what you must never do, or try and steer away from, is use complex verbal language, the technical jargon of science. That's what makes this stuff opaque to them. That's what they don't get. No one wants to listen to. But if you show them the science, they'll get it. But it's just, the trick is, how do you explain it to them verbally? That's really the barrier. I'm going to play a little bit of DNA work, some of my old work from about 8 years ago, but also some current works in progress on DNA. And then also some, if we have time, I'll go into some brain simulations. Now, each of these, the malaria, DNA, and brain, are different scales. That's kind of why I chose them. But there's plenty of other materials, and I'll show you a website at the end where if you want to see other bits and pieces, not only my own work, but all the amazing work in biomedical visualization that's going on at the moment. So, okay. So what I'm doing, and this whole field, is nothing new. Scientists have always created pictures as part of their thinking process, as part of their discovery process, and also to convey what they're discovering to other people, to the public. And I've chosen two well-known examples because they both use technology to look beyond our normal human vision, and then used art to present what they saw. So Galileo, using a telescope, created these watercolors of the moon, and this was a revolution. This is a complete revelation for humans to realize that the moon wasn't this perfect heavenly sphere, but is in fact a rocky, craggy world, a world out there. Ernst Haeckel, who used a microscope to do these exquisite illustrations of the microscopic creatures around us. And for me, this is the lab I came from in my research years. We studied the big round guy in the middle, up the top there. This is Microsterius. This is a single-celled creature, and a little bit self-indulgent, but does lead into the malaria story. This is footage from the lab I worked in, and that was Microsterius, the species I studied. Most life on Earth, almost 99% of life on Earth is all microscopic creatures. We're the exceptions, us big stuff. If you look in the river here and just take a little sample of water, or if you look at the damp soil, a bit of green pond scum around you as you walk down in the gutter there, if you look out into the microscope, it's exquisitely beautiful. And that's what most of life is all about. It's a dinoflagellate, so when you hear about red tides and toxic algal blooms, that was that stuff. This is a sample of soil. So all of these creatures, except that big guy in the middle, these are all single-celled creatures. This is a euglena. That red dot up the front there is actually an eye spot. So it's an alga, so it uses photosynthesis, but it also has an eye. It can tell where the direction of light is, and it can steer itself away or towards it. It's exquisitely beautiful. There's another species. I just had to put it in there because look at that. I mean, there's nothing we can make up as humans that nature's way ahead of us. Cyanura. This is one of the top 3 most beautiful things I've ever seen in my life. Unfortunately, you're seeing it through analog video recording of it, but this stuff is liquid gold under the microscope. It's exquisitely beautiful, and if you know a microscopist who can take a sample of this stuff, I highly recommend it. It is thrilling to see this stuff under the microscope. A 4-cell creature, so we're heading into multicellularity here. Again, this is really common stuff. It's everywhere around us. We're just not aware of it because it's a smaller scale than we're used to. So this is a colonial, a multi-celled species, triangular species of creatures. I mean, how do you make this stuff up? One more and then I'll move on, but it just goes on and on and on. I'd love to just sit here for the next 40 minutes and talk about these guys. This is a diatom, so it grows glass around itself. Each one of those lines is one cell. They get together and they use this form of motility in real time to get around. So like sliding rails, they slide against each other and scoot around. I mean, biology, man. That's how big this stuff, these are giants. These are volvox, very common, 4-celled species, and so on. Anyway, those are all protists. We're going to talk about another type of protist, which is one that actually uses us as its world. That's where it lives. It's malaria, and it not only infects us, but it's also adapted to jump to another species, a mosquito, and also infect that. And so we have many sophisticated systems inside our bodies to fight infections. And malaria, which has been with us, as you'll hear, for a long time, it's developed all sorts of techniques to thwart all of our defenses, our immune system, and to hide from our immune system, but also to use our immune system to infect us and cause disease. So this is the best diagram I know of the malaria life cycle, and it just shows you how complicated this thing is. The main stage you'll hear about is this stage, where malaria infects red blood cells and then bursts out and then infects red blood cells. But there's a whole stage. So here's a mosquito biting a human that's going in the skin, travels in the bloodstream, heads into the liver, and then has an infection stage. It delays for about 7 days and bursts out and then starts infecting the blood. So this top half is all about the mosquito side. I'm going to play an animation. I did this in 2008, and what I wanted to do was do a comprehensive collection of animations that cover this entire life cycle, because I'm looking at this and it's complicated. It is complicated, I can't avoid it, but I wanted to see what all of this life cycle would actually look like if we could see it, and there was a lot of really good data. And I'll show you the sorts of data that I use. So I'm more of a naturalist biologist. I go from observation. Where it's available, I'll take real data, like the protein models that we were talking about earlier today, and incorporate them directly in the animations and also other forms of simulation. But for the most part, I use observation. So this is a scanning electron micrograph of Anopheles mosquito head, and the sorts of things I'm looking at is the detail, these sort of feathery things on the outside of the proboscis. It's sort of a sheath that goes around it. Stylets, these are, they're usually straight. These are the blades I use to chop up our skin. Another one, which is a beautiful summary of how, when the mosquito bites, what I found interesting is, well, the sheath, that feathery sheath, bends backwards, but the proboscis doesn't go straight down. It actually bends around. It's very flexible. And this is actually incorrect. It doesn't enter into a blood vessel. Gruesomely, what the mosquito does is chop up our blood vessels and then lap up or suck up the blood that's spilling out of there. I expected to be a piercing into the blood vessel, but that turns out to not be the case. Okay, so here's the red is the proboscis. This is a mouse's ear that is fluorescently labeled. This is the mosquito's proboscis stabbing in, and this is the malaria parasite, that first stage, that's thin and long, specifically to fit down the proboscis, so which is quite a tiny, tiny thing. It then, also, you might see there's sort of a gliding movement. All of my animations, everything, as much as I can do it, is to scale. The movements are all derived from real-life microscopy, and in fact, there's a couple of moments that look fake, particularly the gliding of the malaria parasite. You'll see it glide around and then glide through a blood vessel and glide through tissue. And you sort of expect it to be, to interact with membranes and so on. No, it just plunges straight through. In fact, it goes backwards and forwards and will destroy cells on its way. That's part of its process. That's what it uses. Okay, and I'll quickly, this is a scanning electron micrograph of blood vessels just under, so this is under the fingerprint, so just under the skin, so you can sort of see these ridges. Those are the lines of the person's fingerprint, and this sort of information gets fed into the animations. I try and reconstruct it. Sorry about the surgery just before dinner, but this is a beautiful footage, and in fact was quite useful in multiple ways. Filmed in Japan. All right, you have the textures. This is the liver, because it's an important part of malaria. But what I found interesting is also the way the heart just through here, through the diaphragm, is sort of thumping onto the liver. I found that sort of dynamics quite interesting. Also, this is looking inside a liver, a transection. This is a hepatocyte, a liver cell, and looking at the blood flow. That was very influential for how I was going to create it, but everything working out things to scale. These little things here are actually beads that they put into the bloodstream, and they've attached to a cell that's sitting inside the bloodstream, which is actually part of your immune system. It's called a dirt cell because it eats all the dirt in your bloodstream, and mainly it's there to absorb all the parasites and so on. It's called a Kupffer cell, and actually that's one thing. For all my animations, when I add narration, I try and never use any technical language whatsoever. I have to find a different way to explain it. This is one I couldn't avoid. The only word I couldn't get rid of was Kupffer, because it is a specific cell we're talking about. So that's the only word that made it into the narration you're about to hear. But the Kupffer cell is specifically there to sense parasites in the bloodstream and eat them. So what does malaria use? But it uses it as a front door to get into your liver. I mean, it's a devious, evil creature. All right, and this is specifically footage from the institute where I work. They're very focused. These are red blood cells, each of these round discs here. And these little guys here are the malaria parasite at the blood stage. And if you watch this one here, you'll see it attach onto the red blood. So they're very focused. The research is focused on how it actually enters into the red blood cell, and if we can stop that somehow, that'd be ideal. We can then prevent or cure malaria or find a treatment for it, or prevention preferably. So it enters, and it very vigorously enters. It reorients itself and enters into the red blood cell. So those sorts of dynamics are all incorporated in the animation. Okay, actually, if it's possible to have the lights down, I'm not sure. This shot here, it's an opening shot. The malaria mosquito, Anopheles, actually bites at dusk and dawn. It's when it likes to feed on humans. This is supposed to represent the neck of a sleeping child. It could be anyone. I meant to make it racially neutral, so because these animations, these specific animations, are not supposed to be used as you're about to see it. They're a resource for Doctors Without Borders. They go and you open up their laptops in a local village and explain what malaria is. The WHO wants to use it in their own types of stories, or documentaries and so on. So this is just a collection of the animations that they can then choose from. It's kind of the idea. But I did add narration because I wanted to have it as a package that go on YouTube or whatever it may be.
The malaria parasite is an ancient organism. It has been with us since before we were human. Famous victims include Alexander the Great, Genghis Khan, and George Washington. The malaria life cycle follows a devious path, swapping back and forth between mosquitoes and humans. This mosquito is infected with the malaria parasite. Because she is pregnant, she has become hungry for human blood. During the bite, she injects saliva to stop the blood from clotting. Her infected saliva also carries the malaria parasite. The parasite rides the bloodstream like a network of roads, seeking its first target, the core of your body's blood filter system, the liver. Sensing its arrival at the liver, the parasite searches for an exit. A sentinel Kupffer cell is the entry point to liver tissue. Leaving the blood, the parasite infects a liver cell, killing one or more other cells on its way. Over the next few days, the parasite undergoes hundreds of nuclear divisions, copying its DNA over and over again. A single infected liver cell can create thousands of new parasites. The next generation of parasites are modified to infect a new target: red blood cells. Inside a red blood cell, the parasite can hide from the body's immune system. The parasite slowly devours the contents of the infected cell and creates more parasites. The infected cell becomes sticky and grips onto blood vessel walls. Once mature, the infected cell bursts, spreading more parasites through the bloodstream. Malaria victims suffer fever, loss of blood, convulsions, brain damage, and coma. This year, 10% of people on Earth will be struck down with malaria. Countless millions have been killed by it. Most people who die from the disease are pregnant women and children under the age of 5.
Not really better news, but good news, but that 10% estimate has been revised downwards somewhat. It's only a mere 300 to 500 million people that are infected with malaria each year. And the estimation is that for many countries, particularly in Africa, that have this just endemic and constant, it actually decreases the GDP by 1 to 2%. So if we were able to eradicate malaria, it will alleviate so much poverty in the world. Because if a member of the family has malaria, they're in fevers and convulsions, the parents have to stay home, or the parents are unable to go to work in the fields and so on. So it's an important one. I'm sorry to start on such a sad note, but I'm going to play the other side of malaria, because the mosquito side of the story, because I think it's actually quite an interesting one, biology-wise. So the opening shot you're about to see is very much homage to this image by Hugh Stark. It's an exquisite, I don't know how he did this, but a photograph of a female mosquito. She's just had her blood meal and launched herself up in the air. Exquisitely beautiful. So you'll see the influence on the animation. And just one last bit of science. This is the stomach of a mosquito, and these structures here are cysts. These are just the sort of forms of information that go into informing and designing the animation itself. So again, it'll be about 3.5 minutes, and we'll be done. We'll move on to DNA science.
Mosquitoes are usually vegetarian, preferring to drink nectar, fruit juices, and honeydew. Only a pregnant mosquito will bite humans, seeking nutrients from blood to nourish her developing eggs. If she drinks blood from someone infected with malaria, she too becomes infected with the disease. The tiny drop of blood filling the insect's stomach is teeming with malaria parasites. The parasite form that is deadly inside humans cannot survive in the mosquito's stomach and is slowly digested with the rest of her blood meal. However, back in the human host, a few of the parasites turned into a different type of cell, one that is sexual but remains dormant. Malaria sex is triggered when the warm human blood begins to cool inside the insect's stomach. The female form of the parasite matures into an egg. The male form takes a while longer to mature into sperm. This sperm is from an earlier feed. The fertilized cell can glide and begins to explore its new environment. It migrates to the outer lining of the mosquito's stomach before transforming into a cyst. Each cyst produces thousands of thin, tiny parasites which seek out and infest the mosquito's salivary glands. The next time this mosquito bites a victim, the malaria parasite will ride in with her saliva and infect another human. This year, 10% of people on Earth will be struck down with malaria. Most people who die from the disease will be pregnant women and children under the age of 5.
Okay. All right, onto the molecular world. So I'm a cell biologist. I love cell biology. And in fact, particularly having seen what's happened the last few days with the presentations of molecular stuff, I think molecular biology is well underway for having some really good solutions for conveying the molecular world to the wide public. But I think, for me, the big task is really cell biology and explaining that one. Yeah, okay, I've got a bias for cell biology. And I used to, this is about 12 years ago, I used to go to lectures by molecular biologists, and they just use this incomprehensible jargon and acronyms, and they'd all focus on individual proteins, and of course they wanted their protein to be the important one, so it was made out, this is the key one that did everything, and I just found it incomprehensible, and I really didn't get it. Until David Goodsell, I encountered his artworks, and this illuminated for me the whole molecular world, the context, the way these things all fit together, the nature of it, the way, what it's all about. And this really changed my career. Just to give you an example of one of his artworks, this is a transection through blood. The yellow-green area is the plasma, the fluids of blood, which is mostly water, and it's got antibodies and sugars and so on, hormones. The red area is a sort of a slice into a red blood cell. The purple is the membrane. The red is the hemoglobin molecules, which give blood its color. And so they are literally like a molecular sponge, and they absorb oxygen from your lungs and then carry it to the different parts of your body. And it was very much these sorts of illustrations that changed my direction. And I started doing molecular work, and for me, it really is an open frontier. It is exciting. It is tremendous. It's mind-blowing, the science that's going on. Probably only second to cell biology, but it's still pretty good. Advantages of using animation is that you can show dynamic systems, and also you can manipulate time, because a lot of the biological actions happen at tremendously small scales, like molecular world, or longer scales over hours or days in the cellular world, so you have to watch things under time lapse. So in animation, you can manipulate that to make this stuff watchable to a general audience. Okay, so this is DNA in its classic double helix form. We can unwind the double helix, and we'll unzip it to see its internal, so its structure. So these things that look like teeth, these are the letters of the genetic code. This is where the genes are written, and this is pretty important. You could ask Eric. That stuff's important. But I want to talk about the mechanical aspects of DNA, which is these two strands, and they physically run in opposite directions, which creates a number of complications for your cells when the DNA is being read, when the gene is being read, or when the DNA is being copied, as you're about to see. So those of you familiar with my work, you're probably sick of this one, but so am I. Let's get through it quickly and get to some new stuff. This is a reasonable representation of the actual DNA replication machine that's in our bodies and all living, and essentially in all living things, or in eukaryotes anyway. The DNA is entering the production line from the left-hand side here. It hits this collection of proteins, with molecular machines that split the DNA and then make an identical copy of the DNA. So one strand, so it's splitting the DNA here, this round donut molecule. One strand of DNA is being copied directly and is spooling off that way. But of course, things are more complicated for the other side because it has to be copied and duplicated backwards. So all of this complicated mechanism is for throwing out these loops of DNA and of a single strand of DNA and copying it backwards. The end result is two identical copies of DNA. Now, there's a lot of simplifications in here, and this was created in 2002, and it was a reasonable representation of what we understood about the DNA replication machine from back then. At the time of creating that animation, things were moving along really quickly, and I've always wanted to go back to that and revisit it and to do a new one. And an opportunity has come up for me to revisit it, and it's a project I can't talk about, but let's just say it's more for the entertainment world than for scientific world. But they did want DNA replication, which is wonderful. And I revisited it just in the last couple months, and unfortunately what I found was the science hasn't moved on, and the issues that were back then are still there. And the solutions I came up with then that seem to fit are still really the only way I can work it out, and it's disappointing. I was hoping that I'd find a review paper and just trash what I had done as a model and say, that was rubbish, this is the way it is, so I could build that. What I found is that there's more bits to it. We're still not sure what's going on, and that's still a reasonable representation. But what I will do as a teaser trailer is show you better dynamics, some better models, and better simulations of DNA. Unfortunately, this isn't going to be for a scientific or an education context in the first place, but we are going to do versions in the future, and I hope to revisit it, and I hope that some of the problems that I had, I'd love to go into that now, are solved by the time I really redo the replosome. So this is more works in progress. It's for a project, an online interactive textbook of biology that we're hoping to get off the ground. Things are looking positive, but we'll see how it goes. I won't go into it again. So if you're interested in biology education, biology textbooks, I'll give you a link to find out more about this project. It's led by E.O. Wilson out of Harvard, and we're currently calling it Life on Earth. So I'm just going to play some excerpts that are all about mitosis and how the mechanisms inside your cells that are used to separate DNA, the DNA once it's been copied. So this is just an excerpt of some of the animations that are available. So again, we begin with DNA, and I've stripped away all that soup of molecules that would be around it. So that wiggling and jiggling is because of the thermal energy and the buffeting of all the surrounding soup of molecules. So I had to strip it away just to make the particular points that I wanted to make visible. So DNA is about 2 nanometers long, but it's about 30 to 40 million nanometers, 2 nanometers across, 30 to 40 million nanometers long. So to keep it organized and to restrict access to the genetic code, it's wrapped around these proteins that I've colored purple here. And all of this is still one strand of DNA. And it's packaged up into this thing, this huge structure called a chromosome. And we'll come back to chromosomes in a minute, where the camera is zooming out, pulling out through a nuclear pore, which is the gateway to this compartment that holds all the DNA called the nucleus, revealing all of the rest of the cytoplasm, the contents of the cell. So we're ending up with a view if you went to the labs here and looked under a microscope at a living cell, it would look something like this. And unfortunately, the processes of a cell occur over long periods of time, so it's under time lapse. The nuclear envelope breaks down, releasing the chromosomes, and you'll see the chromosomes do this very particular and peculiar sort of dance. They line up at this sort of mid-region, which seems to be focused on these little red spots here, which I've added on. And then when the cell feels the conditions are right, it rips apart the DNA. So one strand of copy of DNA goes on one side, one strand of copy of DNA goes to the other side. So both new daughter cells get identical copies of DNA. They're creating two identical cells. I think this version's going to rewind. Yes, so rewind back to this moment, because I'm going to focus on the structure of the chromosome. So that big package of DNA. So again, we're at this moment where all the DNA the chromosomes are lined up at the mid-region. And if we just isolate one chromosome out, we're going to look at its structure, its ultrastructure. All of this is derived and informed by the real scientific data of many different forms of scientific data. In fact, this was the most heavily researched animation I've ever done in my career so far. So these things that look like whiskers are part of the scaffolding inside the cell. They're the microtubules, are part of the spindle, which is usually the focus of this kind of story. But I'm more interested actually in the chromosome. So there's two sausages that make up a chromosome. One strand of DNA is in one sausage. The other sausage is the other strand of DNA. And these microtubules are interfacing with the chromosome through this gigantic red protein structure called a kinetochore. Now, we've known about the kinetochore for more than 100 years, and it's an area of intense study. And if you go to the cell biology meetings, they love arguing about how this thing actually works, particularly about how it moves, how the chromosomes are moving, because they figure that this is actually the center. It is obviously related to how the chromosomes are moving, but I think, anyway. These things that are sticking out on the right-hand side of the microtubules, what you're seeing is the interface onto the microtubule. This kinetochore, this giant, huge protein complex, is a tension-sensing system. It's a broadcasting system telling the cell when it's ready to go for the next stage and separating the chromosomes. You're starting to see it turn green. This is just to indicate that it's getting ready to go and all the proteins are getting ready for the separation. But there's one little last bit that's still remaining red, and that's part of the broadcasting system that it's used to broadcast to the rest of the cell that it's ready. And it's literally walked away by these little molecular machines called dyning that walk it away, mechanically walk it away from the kinetochore, away from the chromosome. So this is molecular eye candy and very much homage to Graham Johnson, but this is orange guys are kinesin who can only walk one direction along the microtubule. Coming along the other way is dyning with that red molecular structure, which is that broadcasting system that's being walked away. There's all sorts of other bits of details, like the blue structures are another type of kinesin that cross-links between microtubules. There's also this wonderful movement of the microtubules called flux, which we have no clue as to why it's actually, they go through the bother of having this movement of the chromosomes, the microtubules. And it seems to be important to do with the separation of chromosomes, but then there's, of course, with biology, there's always an exception. There's various organisms where there's no flux whatsoever, so it's not obviously essential for the chromosome separating. Wonderful mystery. So that's a selection of the animations from the recent mitosis production for Wilson Life on Earth. So I'll show you a website if you want to go check out more of that work. Oh, okay. Sometimes I get really fun gigs. I'm sorry, but this was a whopper, and I just hope it's not the only time I get such a chewy, wonderful project to work on. This is Geneva. The University of Geneva was celebrating its 450th anniversary, and they invited me to, what they essentially did in the very center of Geneva, this road here is the main thoroughfare into the city, bridge, and Lake Geneva is just over here on the right. This is an island on the river, Rousseau Island, and they wanted to transform it into a cell, and I'm completely responsible for the pink. You couldn't miss it, and it's supposed to represent the nucleus of the cell. This was a temporary exhibition. Actually, it was initially supposed to only go for 3 months. It ended up going for 4 months. Open to the public for free so that they could bring families could bring in their kids. From the first week, it was completely booked out with all the school groups and excursions to come in here and experience the interior of a cell. This is their building the model. This is what it looked like inside. So I was responsible for all the projection of the interior of the nucleus. They built this gigantic LED DNA strand, and you could touch the touch screen and it would show you what a mutation looked like, and so on. It was insane. There are all these touch screens around the outside. You could choose different animations to play or bits of clips of footage. This is the animation I created. It's like looking through a giant fisheye lens, so it's very distorted. In fact, this sort of region here was actually about a third of the way, or 2/3 of the way up on the screen, and it was huge. It was absolutely gigantic. And what I hadn't fully appreciated was this DNA here at that scale, so this huge, super high resolution, was about that big across. It was huge, these huge chunks of DNA whipping around the place. And it was actually quite disturbing. You sort of go, whoa, whoa, like this, which I hadn't counted on. But it was a really fun project. And they really gave me a lot of freedom to play and do some wonderful stuff. Actually, this is the main purpose I wanted to show you, is the Swiss know how to have parties. These are the bartenders who are wearing the lab coats. And of course, scientists always have stethoscopes around their neck. You had to find your blood group to go find the shots of drink. you had to eat the gelatinous hors d'oeuvres out of the Petri dishes, which looked like they had bacterial colonies on them. It was a lot of fun. There were all these diplomats and famous scientists and so on there. Yeah, it was an amazing party. And so, yeah, they had 230,000 people more going through it during its 4 months of entry. The idea was that this was a temporary exhibition and would travel the world, and we're going to bring it to the Science Festival in New York and so on. And it cost about €1 million to build that dome, just the dome alone. And it was designed, fortunately for the weather that they get in Geneva, including winds whipping down there. So it turned out that the $1 million that they spent, or €1 million they spent on building it, it was so wonderfully robust that it would cost us about $1 million to take it anywhere. So they've just transported it over to the University of Geneva, and that's where it sits. So it was really disappointing because it was supposed to travel the world, and anyway, the way it goes. So how are we doing for time? Okay. Last bit of stuff I'm going to show you is a completely different scale again, is brain visualizations. Again, a wonderful project led by Mark Ellisman at UCSD. And what they're building is a thing called the Whole Brain Catalog. And the idea is that it's going to be an open repository for all forms of brain data: genomics, proteomics, structural data, tomography, dynamic visualizations of simulations of how neurons function. And the idea is you put this all together as a navigable 3D space so that new things are discoverable. Because the problem is you've got all these different forms of data, and they want to put it all together in this 3D space that everyone will be able to access on their browser, web browser, and to tumble around and you find a cell that you're interested in, you see the context of the other cells, or you may see the proteins that are being expressed at certain times, and so on. Really ambitious. And what they had was seed money to build the prototype, and I was hired to build the pitch piece, the animation, the visualization of what this thing will look like once it's underway. And what they were asking for was a lot of money, and as I understand, I think they tens of millions of dollars, and I understand they are underway and have achieved that. So I should say also, with my animations, there's the sound design, and this one has a musical score, which I really liked. The person who does all my sound design, Frank Tietes, is the guy I do all my sound design with, and he wrote the score to make it wondrous at the beginning. I'm just telling you the story arc. The wondrous at the beginning, to suck you in, to show you a wonderful world, and then to make you open your wallet at the very end. So, okay, forms of data that went into this. And actually, this is the one animation that I'm happy with for the accuracy of it, because it had so many forms of data, and the guys, all the scientists at that were working with Mark gave me so much time. We put in so many forms of data at all scales, so it's a multi-scale data set. I'm happy with what we came out. It didn't end up being the most pretty animation I've done, but technically and scientifically, I'm happy with what the result was. So this is the brain formations of a mouse. So the mouse is it's about the mouse model. What I found interesting, I didn't know this, but the mouse doesn't have these ruffles as we do on the human brain. It actually has a smooth brain. So when you see the smooth brain, that's what it looks like. I had to get data like they did slices through the brain, and this was, in fact, slicing down this direction, but also in different directions. There were about 70 slices that I had to work through, and this is what the slices looked like, because I wanted to reconstruct one particular region, which, so this is the hippocampus here, which is what they wanted to focus on, and then it was this particular layer of cells in the dentate gyrus that they wanted me to reconstruct. We had close-up views of this is just one type of content that went in there, but I had to reconstruct this. I had to rebuild what the whole dentate gyrus would look like. Tomography data, 3D reconstructions informed it. They also had lots of time-lapse movement of the cells. And of course, I went back to the classics. Cajal and his illustrations, exquisitely beautiful illustrations, exquisitely accurate illustrations, observations down a microscope that he did 100 years ago. I'm in awe of this person. He was exceptional. And what I also found interesting, this was a particularly fun moment, because they wanted to show the flow of information through the brain, and I was finding great difficulty in finding how do you know where the information goes, what neurons trigger what neurons. And I was looking at this diagram, and I was like, what are these arrows doing here? And it turns out what they are is actually, Cajal was adding dyes and so on. You could see the flow inside the cells. But I presented to the scientists at UCSD, and I said, these arrows, is this the flow of information through the brain? and from Carl 100 years ago, and they said, "Oh no, 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 that's right." He's done a map of the flow of information. I don't know whether he knew that, but that's extraordinary. So this very much informed the animation, how the patterns. They wanted to also show patterns of behavior. One of the challenges of this sort of work is time scale, and one of the patterns of flow, the cycling of brain cycle you'll see, actually happens at 40 hertz, so 40 times a second. So I had to slow it down to about half a hertz, or one time every 2 seconds, to make it watchable, because I've only got 25 frames a second as I'm working on here, 25 frames, so you have to manipulate time to make this stuff watchable, but at least to give people a sense that there are patterns there. So here we go. We start with a mouse. The mouse comes up and sniffs the camera. The reason we're interested in that is the region we're focusing on is central to interpreting smells, but also brain memory formation. And that's why often smells trigger memories. It's the same region of the brain, or it's the same region of the brain is involved in both. So here are the brain formations. This is the last point where I used real data and everything else had to be interpreted. So I've reconstructed just one single layer of one cell type. Oops, sorry. So this is available in high definition. There's a lot of cellular details that, if you're interested in it, that's available. And so again, we're just looking at one cell type, one tissue layer. This is that cycling of the brain activity. And what's interesting for me is that brain activity is constantly doing a cycle. It's a modification of that cycle that is actually where the information is encoded. We come down, so this is as it is, the virtual environment of the whole brain catalog. You come down, you decide you want to isolate just one cell out of that cacophony of other cells. And this was all based on tomography data, so it's accurate for its structure. You have the axon, which is where the information flows out of the cell, and these are the dendrites where the information comes in. And each of these is different synapses. Each one is touching a different axon. Usually there's tens of thousands of different connections upstream to other axons from one cell, or this particular granule cell. And what you're about to see is the formation of a synapse, the key component of a formation of a memory. All of that wiggling movement is derived from time-lapse microscopy. An astrocyte comes in and tells the dendrite to form a synapse here. We're certainly intimately involved with the formation of a synapse. This is representing the various molecular components that form at the synapse junction. And so the formation of a synapse. But then this project is really about scale and integrating many forms of data and revealing new patterns, new connections, new relationships between all these forms of data. So if you're interested in the continuation of this animation, I too take credit card. Okay, I think I'm close to on time. Yeah, one of my favorite quotes from Steinbeck. After all, I guess it doesn't matter whether you look down through a microscope or up through a telescope, as long as you look. And I think that's actually key. We need to really go back and look at biology. Look at what living things are actually doing. Before we grind them up and look at them in many little tiny ways, we really should see the big patterns, what's really going on, particularly in cell biology. And finally, if you want some more stuff, WeHi.tv is the repository for most of my work, also on YouTube. MolecularMovies.com is Gail McGill's collection of the best of all sorts of biomedical animation. his curation of that kind of work. So I'd highly recommend that, and also you can get Molecular Maya from there. Wilson LOE is the biology textbook, the prototype chapter, which we're raising funds for at the moment. And the whole brain catalog is did I spell that right? Yeah, is the brain catalog. So yeah, thank you.